So we're happy to have with us today a friend of the Causal Inference Working Group, Peter Steiner of the University of Maryland. He researches in the Department of Human Development and Quantitative Methodology, which is under the College of Education, and his research focuses on, within causal inference, focuses on replication and survey design. And I'll hand it over to you, Dr. Steiner.
Okay, thank you very much, Brian, for the introduction, and thank you very much for this invitation. I'm going to share my screen now, but please feel free to interrupt me whenever you have questions. I'm happy to take questions throughout the entire talk. So I will talk about a causal replication framework, particularly the assumptions and some design aspects. And this is joint work I'm doing together with Vivian Wang from the University of Virginia. And we also got some grant support by NSF and IES to do this research. Okay, so I think you're all familiar with the replication crisis, and you read about something that the psychologists declared a big crisis, that they failed to replicate results. You might have read about sloppy sciences. And this is not only typical for psychology, but there were failed replication efforts in economics, medicine, education, prevention sciences, so in the whole range of applied research. There were also quite influential papers, for instance, by Ioannidis, why most published research findings are false, or by Benjamini, who says it's selective inference that is the silent killer of replicability. Right? But when we have a closer look at the causes of the replication crisis, there are some aspects that ranked very high up there. Like many people, particularly in psychology, think everything is to be blamed on significance testing or the related power issues, right? But then close up, there is some issues with publication bias, questionable research practices, researcher degrees of freedom, the guard of forking paths. So essentially sloppy science here. But what is not that often discussed is, it could just be that our knowledge or some of the data generating processes out there in nature are just not as stable as we believe they are. So there might be quite a bit of effect heterogeneity across populations, sites, times, but this issue of effect heterogeneity rarely get discussed. And there might also be a lack of a rigorous formal definition of replication is. And I will talk a little bit about this, how most approaches to replication think about what replication is. And there is still a lack of optimal and practical replication designs. So there's replication is not yet considered as a research question on its own. Therefore, people don't think that much about implementing strong replication designs. Instead, there are many post hoc replications without lots of systematic thought behind it. Therefore, it's not surprising that we see a replication crisis. The problem, in my opinion, is not that we fail to replicate previous results. The problem is that we do not know why we fail to replicate results. Right, and the final point is there might be a lack of agreement or guidelines also how we assess replication success. So there are lots of measures out there how we should do this, but also depending on which measure you choose, you will get very different results. I'm not talking today about the choice of appropriate replication measures or correspondence measures, but we did a reanalysis with the OSC data, and they claimed about some 35–38% of replication success. So when we use a correspondence measure that merges significance testing with equivalence testing together in one test, then we find that for about 70% of the studies, the sample sizes are too small to be conclusive to begin with. And of the 30% remaining, there is about half of them, about 20% is a replication failure, and maybe 10 might show an equivalence. So the other problem is, as I would argue if I would give a talk on different replication measures, is that almost all those tests for assessing replication success are hopelessly underpowered. Therefore, we better conclude there is not a crisis, there's just a lack of sufficient evidence to come up with a conclusion whether there is replication success or failure. So, given this lack of a strong research design, so Vivian Wong and I, but also Larry Hedges and Jake Schauer, think that there is a need, what we could call for replication sciences, or think about replication as a research design. So what this requires is first a clear definition of what replication is and what the underlying assumptions are to better understand issues related to replication. Then we need to think a little bit more about strong research designs for replications that allow us to learn from replication failure particularly, right? And that allow us to deal with the uncertainties out there in the changing world. And then also we need to think about the appropriate analysis. What are the appropriate correspondence measures? We need to think about what role meta-analysis can play here. And of course, open science and transparency efforts are very important. I will allude later to it because we really need to have access to the raw data of the original studies. Otherwise, it might sometimes be very difficult to do an appropriate replication analysis. So what will I talk about today? That is, first, what is replication? Then I will introduce a causal replication framework using potential outcomes notation that allows us to clearly define causal estimates and then derive the assumptions that are required for direct replication. Since direct replication is really of interest in science, we can nonetheless use this framework to derive research or strong research designs that allow us to systematically vary factors of the replication studies. And then I will conclude with implications for practice. So what is replication? I have here a very nice quote by Schmidt published in 2009, where he defines replication is a methodological tool based on a repetition procedure that is involved in establishing a fact, truth, or piece of knowledge. Interestingly, most researchers read replication or interpret replication as a repetition procedure rather than establishing a fact, truth, or piece of knowledge, right? So I will show you a few definitions of what replication is, and they all focus on repeating an experimental procedure. And that's what is typically called direct replication, sometimes also exact or close replication. So here are three quotes, but there are many more of those quotes that highlight it's repeating a methodology. That's what replication is about. OSC defines it as recreate the conditions, although they are not as explicit, but mostly mean also that you should use the very same randomized experiment, instruments, settings, populations in order to establish a replicability of the effects. Also here very explicit by Brandt et al., methods and procedures as close as possible to the original study should be replicated, then we will call it a close replication. However, there are some issues with replicating methods and procedures. First, such a repetition procedure prioritizes the original study over the replication study. It's implicitly assumed that the original study identified a causal effect and that there were no flaws associated with it. That's the second part. What if the original study has some flaws? Let's say there was non-compliance in the RCT, there was attrition, there was low treatment fidelity and treatment contamination. And in psychology, people don't worry that much about a little bit non-compliance, a little bit attrition. They just analyze the RCT as if it were an RCT, and that replicates particularly the procedures they implement. They implement randomization, and they do this, and when there are some flaws to it, they might report about this, but they typically consider them as negligibly small. But whether they are is a different question. So the original study might actually be a false positive finding. And of course, it does not really make sense to start replicating imperfectly implemented or flawed studies. And replicating a flawed study might not be very useful or meaningful to begin with. The other issue with replicating methods and procedures is that all the procedures are rarely fully documented in the original study. Often researchers might not even know what are the procedures that are important with regard to the replication of results, meaning which procedures or settings actually affect moderators, and they would need to be reported. So that there is, I got confused because my screen was blinking a little bit. Okay, such that there is, due to the lack of documentation, it's not possible to have exact or close replication if we have a lack of this documentation. Then one of my major critique would be that replicating methods and procedures is not the primary goal of a replication study. As already said, we would like to replicate a fact, truth, or piece of knowledge. And another argument made by Hedges and Shara is that methods and procedure definition of replication is not precise. There is no specific criterion what replication actually means. So it is difficult to optimally design a replication study and also to think about what's actually a good analysis to assess whether we have replication success or failure. Therefore, what we did is we mostly focused in defining what replication is on establishing a fact, truth, or piece of knowledge. So what we say is what we would like to replicate is not the methods and the procedure, but a causal effect of a well-defined treatment control contrast in a specific population setting, which we then refer to as the causal estimate. Nonetheless, in order to achieve this goal to replicate a causal effect, it might still be useful to replicate some methods and procedures, because this might actually help in achieving the replicability effort after all. But we do not require it, it's not really necessary. Okay, so here's the causal replication framework for defining the causal estimate. So we take a causal point of view, right, instead of a procedural one. So it does not necessarily need to be a causal effect. You could also focus on associations if you like, but it doesn't become really easier because let's say there's some confounding issues, then you would need to make sure that in both studies, the confounding happens to the same extent in addition to the causal effect. So which is not necessarily easier than looking at causal effects to begin with. And we do not prioritize the original study. We think of from a prospective point of view. What would we do if we plan a replication study, in the simplest case with only 2 replications, and think about it as a research design? Okay, how would we later assess replication success? How large do the sample sizes need to be in order to have sufficient power to establish equivalence of the effect estimates or differences? Is there any question? Okay. And we use the causal approach to derive assumptions that are required for a successful replication of causal estimates. So just to review again, and that's very crucial, how we define a causal estimate. So we see it as a population parameter that quantifies a causal effect, but it's important that it has to refer to a well-specified treatment control contrast. So it's not only the treatment condition, it's also the control condition, and I will come back to this later. It needs to refer to a well-defined inference population, which I will abbreviate with R, and it needs to refer to a well-defined setting S. Okay, and for this treatment control contrast for a well-specified inference population and setting, we would like to estimate the true but unknown causal effect in both studies or in multiple studies. Okay, so I use potential outcomes notation to formalize this a little bit, but I derive the potential outcomes from a structural causal model as it will be helpful for the discussion. So let's assume there are two studies, a study A, and I use subscript I for the subjects in study A. Then with a study B with subject index J. So the studies might be belong to the populations might be different. There might be different settings across studies, sites, and times. So
sorry, could you explain a bit what you mean by the setting? Like the population, the intervention makes sense. I'm just not familiar with the idea of the setting.
Right. The setting is just so, let's say you might do a study in a lab. You might do it out there in the field. When you do it out there in the field, you might measure people at home. You might measure them at the clinic, right? And all these decisions in which setting it is, and in which environment, you can also refer to it as environment, might affect the measurements. Right? So therefore, so in different settings, lab settings, field experiments, let's say you measure patients at home, you measure them in a clinic, and all these things might contribute to different outcomes, right? And they could say different sites is part of the settings. You could also say measuring at them different times is part of the settings. And I will formalize the settings, including also sites and times later on in S, as we see here. So the data generating process for the outcome Y can be conceived as a structural assignment. So the potential outcome in study A is just a function of the treatment in study A, then the subject characteristics X, and the setting characteristics S, which includes now also different time points, maybe different sites, might refer to different measurement time points, timing issues, and all these things. Okay, so that's on the structural assignment in order to generate the potential outcome. So I have a pair of potential treatment and control outcomes here for the study A, and I have a pair of potential treatment and control outcomes for study B. And the only difference is on control condition, treatment condition, control, and treatment here. Okay. So the first thing we have to do, we have to define a causal estimate. Let's assume study A implemented and study B2 implemented a randomized experiment. Let's assume study A was perfectly implemented, no attrition, non-compliance issues, and so on, such that they can identify the average treatment effect. And it's the average treatment effect with regard to study population A, setting A. And in order to make it explicit, I take the expectation over the study population A, and also here I have the definition conditional on the setting. In order to make it really explicit that this is a definition of the average treatment effect, the conditions on the setting in which we implemented the study. Okay, but there might have been issues in study B, there might have been non-compliance. So they already face three different choices. They might estimate the intent to treat effect. They might estimate the average treatment effect on the treated in case there was only one-sided non-compliance, meaning there were only no-shows but no crossovers. Or if there was two-sided non-compliance, they might look at the complier average treatment effect. Right? And we already see here, if we presume effect heterogeneity, those effects, the complier average treatment effect or ATT, of course, are likely to differ from the average treatment effect. Right? But in order to succeed in replicating the two studies would need to look at the same causal estimate. But if they start looking already at different subpopulations or different quantities, ATT versus ITT, we should not expect that we see replication success. Okay. So we can expect a successful replication only if both studies, let's say we look at the average treatment effect, if both studies look at the same causal quantity, right? However, they still might have different populations and they might still be implemented in different settings. And they are typically implemented with different samples and different settings. Okay, so in order to derive the causal replication framework, so as I said, we use a prospective point of view, and we will ignore all the issues other people discuss, like publication bias, p-hacking, or hacking hypothesizing after the results are known, right? And we just focus on the question, let's assume there are no issues like this out there. Will we even have a chance to, or have a high probability of seeing replication success? And here we only focus on identification and a little bit on, not really an estimation, we focus on causal identification and ignore all other aspects in this talk, at least how we assess replication success or failure. And we are not talking about power issues as well.
Peter, can I ask a question?
Sure, please.
So let's suppose that RA and RB overlap.
Yeah.
And SA and SB overlap.
Yep.
And I could possibly estimate using data from study A and study B what the average treatment effect would be for the overlap population, right?
Correct. You could do a reweighting.
And that might be a measure of replicability.
Yeah, correct.
Right. Is that something that you consider, or that's not really what you're?
So in the definition, it's not of relevance, but as you will see in one of the following slides, if you have different populations in different settings, and assuming we have enough variation in the settings, of course, you could reweight the study populations in setting to a well-defined replication population and setting scenario. You can do so. And what you need to do is just to make sure that the causal assumptions are met, that the effects are transportable to your replication population and setting.
Correct, yeah.
Right. But that's exactly what people should do. And that's why we need to have access to the raw data of the studies. Otherwise, we cannot do this reweighting, right? And in almost all the studies, the population characteristics will differ, such that we need to do this reweighting in order to have a chance to look at least at comparable populations.
So I'm just thinking that 8, the way you've defined it up there, they might not be equal, but it could be the case that when you reweight to.
Yes, exactly.
That they are equal.
Yeah, so right now at this basic definition, I'm more restrictive. I'm still allowing some differences, right? I'm just requiring this here. And now I will talk about under which assumptions this will be established. Okay, so just one slide that there are alternative approaches, particularly suggested by Larry Hedges and Jake Schauer. They formulate what replication is and what we want to replicate in terms of meta-analysis. And so their quantity of interest is the meta-analytic effect size parameter theta that represents the quantity of interest for replication studies. But I would say, so it's very hard often to say whether this effect size parameter really warrants a causal interpretation or not. It depends just on the studies you use for the meta-analysis. The other issue is you need to then use subject matter knowledge to decide what's a tolerable variation in effect size, such that you still will conclude that there is a replication success. And it was criticized already by Mather and van der Wiele about that it's not necessarily a satisfactory approach from a causal point of view as using meta-analysis to assess replication success. I would essentially agree with this, and meta-analysis makes sense when you have multiple studies. With 2 studies, you're not yet in a good position to begin with. But if you have lots of studies, right? Then of course, I would say so meta-analysis is a good way to do so in addition to pairwise comparison of effect estimates. Okay, so here's the causal replication framework that Vivian Wang and I suggested. So we have two sets of assumptions here. One set refers to the replication assumptions, on which assumptions need to be met such that we can guarantee that both studies look at the same causal estimate. So this is assumptions that need to hold across studies. And then of course, we have study-specific assumptions. We need to make sure that for each study, we can actually identify and estimate a causal effect here. In order to claim that we actually comparing causal effect estimates here. So now here's the list of assumptions. That's just the overview. There are lots of sub-assumptions behind those. So the replication assumptions refer to treatment and outcome stability and the equivalence in causal estimates. And then we have the study-specific assumptions that the causal estimate must be identified in both studies, must be estimable without bias in both studies. And then finally, there must be correct reporting, which then goes into open science and transparency efforts to have everything reported in sufficient detail. So here is the first set of assumptions referring to the treatment outcome stability. So the most important thing I would say is that there's no variation in treatment and control conditions across the studies, which means we need to have identical treatment procedures in place in study A and B. And also the control condition must be the same. Frequently people use business as usual control conditions, but if the two studies differ in their control condition, we should not expect replicability. If we want to formalize this, it's rather easy to see if I look at study A. So if an individual in study A would not get the treatment as implemented in A, but the treatment as implemented in B, then we need to have equivalence in the potential outcomes. The same, of course, if a subject exposed in study B exposed to the very same treatment as in study A need to have the same potential outcome, right? Then we have no variation in outcome measures, right? So here now it varies on how we measure the outcome Y. So if I measure the outcome in study A, I should get the same outcome in study, I should get the same potential outcome if subject I in study A would be measured with the instruments in study B, right? So the two studies might use different instruments, which is perfectly fine as long as they look at the same construct. Right? In order to make a problem better clear, we should put expectations around here such that there might be individual variability. We do not necessarily require exactly the same potential outcomes, but that's what we require, right? The important thing is it not only implies the same instrument, it implies that we measure in essentially comparable setting and timing. So if we measure in different settings, or if we have a different timing, meaning that we don't measure the same time after the treatment implementation, if this makes a difference, then of course we get different measures and we might not necessarily expect replication success. Then next assumption, no mode of study selection effects. How you get into study A or study B should not make a difference. Right? For instance, study A might use incentives, study B might not use incentives. So in study A, people might be more motivated because of the incentives. So what we need to rule out is when we have G, might be factors here, how you get recruited into study A and into study B, that those factors are not of relevance, that we can drop this from our notation. Right? Then we can rule out mode of study selection effect. Right, we have similar assumptions then, of course, within each study, as you know, with the stable unit treatment value assumption. And that exactly mimics the stable unit treatment value assumption at the study level, not at the treatment and control assignment level. And then we need to assume no peer spillover and carryover effects, which means when we conduct study A, the potential outcomes of the participants in study A are not influenced by characteristics of study B. and the potential outcomes in study B are not influenced by characteristics of study A, either research influenced or participant influenced and so on. Okay. So this is already a pretty strong set of assumptions, but we are.
What about secular trends? So if study A is conducted in 1990 and study B is conducted in 2010, and there are changes in medical care, how is that reflected in your assumptions?
Coming up now. We are just at the beginning of the assumptions.
Oh, beginning. Okay, good.
Yeah, we are just at the beginning. There are several slides following because we just started. Okay, so the second group of assumptions is equivalence of causal estimates. So we have the same causal quantity of interest. This is rather straightforward, as I said before. Both studies need to look at, let's say, the average treatment effect. It's not possible that one study looks at the AT and the other at the ITT or the complier average treatment effect. Then we know that we look to begin with at different populations. So then we have the second part of the R2 assumption, and that's probably the crucial one. That's what you were alluding to, identical effect generating processes across sites, times, and so on. Right, so what I do here is I rewrite my potential treatment control outcome for study A as a function of the potential control outcome plus an effect generating function tau A. And tau A no longer does not necessarily need to depend on X and S, meaning the overall set of variables, but what I denoted here with the dot series, these are only the effect moderators. Right, so it's sufficient to have the effect moderators in there, right? And what we require if we have studies conducted over time is that the effect generating process in study A and study B has to be the same, right? Meaning so these functional relations tau A and tau B have to be the same, because if they change over time or if they vary over sites, we would not expect that we would successfully replicate the causal effects. Okay, so this was only with regard to the tau A and tau functions. Here now we need to look at the X distribution and the setting distribution. Okay, that's formulated in the third part of this set of assumptions, identical distribution of population characteristics. So which just requires that, if I go back a slide, we need to require that the distribution of the effect moderators in study A and study B are identical. And then the next part of the assumption would require that the distribution of settings in study A and study B are identical. And we only require so with regard to the effect moderators. If they would differ with regard to, let's say, idiosyncratic causes, it wouldn't really make a difference. Okay, so what I write is if we have different distributions of effect moderators in our studies, in study A and study B, then of course we would no longer get the same causal effects in study A and study B. The same, of course, if the setting distribution differs, then we also can no longer expect that we can replicate the causal effects. Okay, so these were the assumptions across the studies. Now we can look at the identification assumptions that need to be met for each individual study. And let's just look at an example. Let's have an RCT with RCTs with identical target populations and settings in case there's perfect implementation. Right, this would be the perfect setting, and you can relatively easily establish this by randomly assigning participants into study 1 and study 2. Within study 1, you randomly assign them into treatment control condition. Study 2, randomly assign them in treatment control condition. It would be a doubly randomized study, but it's not a preference trial because in both studies you would have random assignment. In there. But that's, as you realize, that's usually not what we do. That's not really of practical interest unless you would randomly have one study is done at clinic A, one study is done at clinic B, then you would need to first randomly assign participants to the clinics, and you could think about something that's actually useful to see whether there are variations due to the settings, meaning the clinics and maybe the physicians working in the clinics or the methods available in the clinics. Okay, let's consider a more realistic scenario where RCTs have different target populations. And we briefly talked about this. The target populations of study A and study B might be different, also the settings, right? And let's assume there was perfect implementation. Then, as we said, we could define a replication population, which might be RA or could be RB, but it might be the average of RA or RB if we like. But then we would need to reweight the populations with regard to the effect moderators. And then we could recover in each study the replication population of interest. The same can be done with the setting, provided we have in each study A and study B variations in setting and that the settings overlap. But often the settings are fixed for a study such that we often cannot match on the settings because there is often no overlap. That's the problem with differences in settings. Then the second set of assumptions with regard to the unbiased estimation of causal estimates in each study, which means you need to choose, once the effect is identified, either an unbiased or consistent estimator for the average treatment effect, if the AT would be the estimate of choice. And you need to have unbiased and consistent estimators of the standard errors that reflect the uncertainty in your designs. Otherwise, you might get invalid significance tests and draw incorrect conclusions about replicability. And of course, technical assumptions like no perfect collinearity, etc. And finally, it's important to correctly report the estimates, the estimators, the estimates, such that you can make the results replicable, such that other research teams know what was the target population, how did the target population look like, which estimators do you use, and so on. Okay, so these were the set of assumptions. And as you might already realize, they are pretty strong in most cases, these assumptions, such that it's often not very realistic to expect replication success to begin with. So just to show you a few examples, and this would be the hypothetical case where we have two RCTs. And let's assume participants get randomly assigned in study A and study B, such that we know the populations equivalent. They might get implemented in the same setting, such that populations are identical, settings are identical, both studies, the AT is identified. This would be the ideal scenario. But this is kind of a boring scenario from practice, because in such a setting, we would expect replicability. It's just doing the very exact study twice, unless you start introducing systematic variation, as we will see then later. Okay, but more realistic is that we have a study A, might be an RCT, study B, an RCT, and that one study might have used incentives and it might have affected the potential outcomes such that we get issues in study A here. Then there might be different replication populations R, there might be different settings S. And if we believe that the differences are effect moderators, then we could not identify the ATE with regard to the replication population in study A, because I defined the replication population as the population in study A and also the setting as in study A. But study 1 fails due to incentives affect the potential outcomes. Study 2 fails due to the non-equivalence of settings and populations. Okay, so that's a more realistic example, but you might be able to do something with regard to reweighting the populations. As I said, might be a little bit harder to deal with this effect. But you don't need to use two RCTs. You could have a lab RCT and later you want to replicate the effects in the field. But then here a major problem might be that you no longer have that much control over the control condition, that you have a different treatment control contrast, even if the treatment is exactly the same. Then you might have different timing of the measurements. It's much easier to do measurements in lab conditions than in field settings. You might have potentially carryover effects because same researchers might do the field study, they might have learned from the lab experiment and change what they are doing. Then we might have different effect generating processes because in a lab, they are frequently not identical as we observe in the field. So there might be other effect moderators out there with different influences on it. And we have different settings due to the lab and the field experiment, such that we would not expect that the field experiment would replicate the lab findings, right? So it would be a replication failure here. Okay, so this was all about direct replication, but frequently it makes sense not to even implement a replication study that aims at exactly replicating the same effect. So what instead we would do is we would systematically vary some of the factors, which means we would systematically violate one, ideally only one, but potentially more assumptions and have planned design variations in there, while all the other assumptions should be met. And that's typically called as conceptual replication, that we have systematically planned variations in one of the studies. Then if we only vary one factor at a time, right, we should be able to learn from replication failure, right? Because we then know the cause of why we failed to replicate the effects. If we varied only one factor in that, but that's of course difficult to do. Often there are 2 or 3 factors varied simultaneously. So what we like to do is if we observe effect heterogeneity, we would attach a causal interpretation to the effect heterogeneity. And this also helps us to generalize causal effects potentially to other populations then. So this would be a setting where I have 2 RCTs and I try to keep everything constant, except let's say I just vary the replication population, but do everything in the same setting. So only one factor is varied, resulting in a violation of a single assumption, while I try to maintain all the other assumptions, which allows me to attach a causal interpretation if we have replication failure. Or you might just vary the settings, like is frequently done in multi-site RCTs, right? Because in different sites, you typically have different settings. Or you might vary the treatment condition, but still using the very same control, or have different components in your treatment and try to evaluate different components here. Or as what I did with my colleagues when I started getting into this field is on having RCTs comparing with observational studies, trying to keep everything constant. The only thing that we varied is one study was an RCT, the other observational, and we were interested in the question, is the unbiasedness assumption, the unconfoundedness assumption met with the observed variables in the observational study. So it's an evaluation of research designs, whether they can work in practice. In the health sciences, you might think of a doubly randomized clinical trial where you're interested in getting a preference effects, right? Where you have also randomly assigned participants into an RCT, into an observational study with self-selection. And if there are differences between the 2 effect estimates, you would attribute it to preference effects. Okay, so the goal of such a systematic or conceptual, systematic variation or conceptual replication efforts is mostly some probe generalizability and effect heterogeneity. And that's what we typically should do much more in order to learn from replication failure and build a stronger theory about what works under which conditions. I also gave an example that we can use them to evaluate research designs like matching designs from observational studies or differences designs and see whether we can reproduce the effect estimates from RCTs. But you can also use such studies to evaluate the properties of different estimators, but you could use here the same data from a single study. You don't necessarily need to have two different studies. So let me talk a little bit about possible research designs, and then I'm more or less at the end of my slides. Of course, we know this for a long time. People used factorial RCTs since the 20s, essentially, and blocked randomized designs where the blocks might refer to different sites, such that we actually systematically do replications. And replication, since the beginning, was a crucial or a key ingredient to randomized experiments. So nothing new from the RCT side in here, and multi-site RCTs or cluster randomized, multi-site RCTs are also very commonly used in different fields of research. However, some people criticize those studies because they are done by the same research team. What they would like to see is have an independent replication that, let's say, one research team does the implementation analysis in site A and another research team does it at site B, completely independent. Right? This is possible for multi-site RCTs. It might get a little bit trickier for factorial or blocked RCTs to split it up into independent research efforts, but it's possible, I would say, under some circumstances. But a more realistic in practice, there are randomized cohort designs where you just stay at the same site and you just repeat your experiment 1 year after the other. Right, but having a new cohort, let's say, of students coming in, and you essentially rely on the assumption that there is no change of the effect generating process over time, right? But you could hold everything else constant and try to see first whether there is a change over time or not. And later you could start varying other factors in order to systematically learn what makes a difference. Similar designs, of course, switching replication designs, where you first randomly assign participants in treatment group and the control group. And after this is done, you switch them. The treatment group becomes the control condition becomes the treatment condition. Rests, of course, on some assumptions here that, let's say, the treatment conditions, treatment effect goes away, or if it does not go away and maintains at the level, then you would try to replicate the pattern that the control group, after receiving the treatment, would catch up with the treatment group with regard to the outcome of interest. Similarly, there are stepped wedge designs where you have not enough resources to expose everyone at the beginning to the intervention. But you would first have, let's say, a randomly selected group of participants that get the intervention. And after the intervention was implemented, then a second randomly assigned group will get the intervention. So it unfolds over time, the treatment implementation in steps or waves. And you could first try to directly replicate the effects because you have to have differences in time. So you might want to see whether there are whether the effect generating function changes over time. But once you establish that there's not really a change, you might think about relaxing other assumptions, using different treatment components, applying it to different populations, and so on. And ideally, you combine those designs into a stronger study where you have multiple groups in different cleverly thought out combinations of design elements in there. And Vivian Wang and her colleagues is currently implementing such a study in an effort to systematically rule out most plausible threats to validity in a replication effort here. Okay, the important thing is a meaningful interpretation require ideally that you only vary one thing at a time and not everything simultaneously, which is difficult to do, of course, in practice. Therefore, it might make sense to have multiple replications simultaneously going on and that they jointly produce a pattern that allows you a causal interpretation. And again, repeating methods and procedures might help keep them stable if necessary, but you don't necessarily do it if there are good reasons for it. Okay, maybe I just said all these things, just let me skip to the last slide here. So we would claim that it makes sense that you plan replication efforts prospectively and not retrospectively when there's one study out there and then you use the study result to replicate the findings. It's good to do so prospectively. And for instance, the Institute of Education Sciences is systematically funding such efforts. So there will be funds out there where we can do this. And interesting, I learned that IES would be even willing to fund replication efforts where different research teams, let's say, submit 3 proposals separately, but they all look at the same causal quantity of interest. Let's say they might differ in target populations, a little bit different settings, but they would allow that 3 independent research proposals are submitted with regard to the exactly same topic implemented during the same time, but coming from different researchers can make it, provided each study makes it separately through the review process. And that's a little bit the tricky part here. So then the important thing is we need to have the data available because we might need to do some reweighting with regard to the population or look at the subpopulation only, whatever we find makes the most sense. And when we use design variants, we should think hard to systematically vary only one factor at a time and not to relax everything simultaneously. And that was typically done in the collaborative efforts, for instance, of the open science collaboration, where the studies are very timed, they're very populations, they vary probably the control contrasts and not only the treatment contrasts, though there were many issues involved to it. And pre-registration of replication efforts and following transparency guidelines might be very helpful to do so. And I think we need to really do replication studies out there in practice and do them in a systematically planned way to show that we actually can build stable knowledge about which intervention works for which populations, under which settings, and whether what worked 10 years ago, will it still work today? Okay, thank you very much for your attention. Any questions?
Thanks so much. I guess I have a question to start. So replication, I feel like, is the replication crisis is basically talked about in the social sciences, but the mathematical assumptions that you presented could sort of be applicable to any kind of science, because basically all fields of science have a need for replication. So I was wondering if, I mean, it's in a way it's sort of intuitive, but I was wondering if you could explain like why in social science replication is more problematic and whether there's anything to be learned from looking at other fields of science, like physical sciences.
Right. So, I mean, so in the social sciences, and you're absolutely right, in social sciences, we don't have, let's say, that much stability in the effect generating functions. Everything changes. Let's look at the pandemic, right? Everything changes in educational research. And also with regard to medical outcomes, people are more likely depressed. So we can not really compare results from before the pandemic, during the pandemic, and probably quite different after the pandemic. Right? But in the physical sciences, they have much more stability in there. And that's where the notion comes from, that we want to uncover stable knowledge. If we cannot find stable knowledge, then maybe we shouldn't call it science. That's what some people might actually claim in the hard sciences here. But there's definitely things one can learn from the different fields with regard to significance testing, standards for implementing these things, and people look at those things. Yeah.
Any other questions?
I have one question in observational study perspective. In observational study with a similar population structure or similar setting, bias is also likely to replicate over studies. So is there any way we can reduce that flawed replication of bias across studies? Like such as by diversifying settings or diversifying the source of bias across different studies, so on.
All right. So the first important thing is that people would like to have very different independent research teams to implement the study. Because if you have the same research team, you very likely replicate the same flaws, right? So, and if you have a fresh research team that might not really know how the original research team implemented the things, might actually help in uncovering biases, right? And one could look at the protocols. What did the research teams do differently? And this might give you an idea on where the effect heterogeneity comes from, right? But there might be, again, many sources of effect heterogeneity out there, right? So in planning replication efforts, the difficult part is finding a balance, what to vary, what not to vary. Right? When you want to have a completely independent research team, you cannot keep them completely blinded what you did in the first study. You need to inform them. The question then is, how much should they know and what should they not know? How much of the research protocols should they know? How much do we want to share with them? Right? And I would say it depends on your research question. What's the target of a replication effort? Which aspect of effect heterogeneity would you like to investigate? If you presume there is hidden confounding somewhere, you can design a study to look at this question by holding everything else constant, right? I think you need to have a clear list of priority what you want to do with the replication effort and not just go out there and do an unsystematic replication because we wouldn't learn anything.
That makes sense. Thanks.
Any final questions for Dr. Steiner?
Is there, money in these different fields, social sciences, to actually do the replication? So have you found that there are funding agencies willing to pay for these kinds of replication activities?
Yes, they do. For instance, particularly IES has a strong effort in funding replication. And as I said, even if there are two studies coming in doing the very same thing, both of them can get funded, right? The only thing that is a little bit lacking is that it would be great that there would be a little bit coordination between the two studies. I mean, they're usually coordinated because you have similar consultants on there and other people on there, that you see that they shared their thoughts, right? But it would be better if they would explicitly say, we are explicitly funding sets of 2 or 3 studies conducted by independent research teams, but with a joint coordination effort. That's what I would like to see. And I mean, IES is going in this direction, and they have an own line of replication studies to be funded. And it's already now in the 3rd round or 2nd or 3rd round that they fund replication studies, and they take a huge emphasis on replication.
That's great.
Yeah, and also that the publications, the outlets, encourage replication efforts to be published, of course. So it goes hand in hand. So it's getting better over time, I would say. But there's not yet enough knowledge about how to best implement replication efforts. Still, when I review these proposals, it's very unsystematic. They vary everything simultaneously. And then when we fail to replicate, we don't know why, right? That's the major problem here. Still is. So it takes another 1, 2, or 3 years until people learn about replication designs.
Thank you. Very interesting talk. Thank you.
Thank you very much.
Oops, sorry, I was muted. All right, thank you, everyone. And thank you, Professor Steiner. I'll see you next time.
Yeah, I thank you very much for the invitation. Have a nice day.
Thank you.